Hello. This is going to be chapter number 3. We're going to talk this chapter about cellular level of organization. If you remember, in previous chapters, we were talking about the atomic level of organization and molecular level of organization. Both of them are chemical level of organization. Then, if you remember, a group of biomolecules going to be organelle, and a group of organelles it's going to be the unit for life, it's going to be the cell. Then we're going to talk about the cellular organization, the cellular level of organization. A cell is going to be really different in shape and function. We have trillions of cells with different shape and function, but even so, a generalized cell can be described with three specific parts. They're going to be plasma membrane, it's going to be the nucleus and the cytoplasm. Then again, every single cell in our body, every composite cell is going to have three parts: the plasma membrane, the nucleus, and the cytoplasm. Then the plasma membrane, the main feature is going to be the semi-permeability. That is going to be selectively permeable, and it's going to be because they are able to regulate the passage into and out of the cell. The control center of the cell is going to be the nucleus. And inside of the nucleus, we have the nucleolus. It's an organelle which has specific functions, and we're going to be talking about that later. Sorry about that. And some cells, some mature cells in our body, such as the red blood cells, they don't have nucleus. Then they are called enucleated. And some other cells could have more than one nucleus, and they are called multinucleated. And finally, we have the cytoplasm, which is the matrix, the gelatinous semi-fluid matrix of water and suspended and dissolved substances in there. Then you can check here, every single cell has a plasma membrane. And that's a plasma membrane is composed of phospholipids and protein molecules, regulating the entrance inside of the cell and outside of the cell. Is the permeability is going to be semi-permeable. The plasma membrane is said to be selectively permeable because, again, it's regulating what goes through, into, and out of the cell. Then we have the nucleus. Right here, this structure is the control center of the cell. And inside of that, we have this matrix. And we have two parts, a liquid part, a fluid part, called cytosol. And we have some solid organelles, as you can check here, these membrane systems as well, they are solid. And they are called organelles. We have here another graphical representation. This outer cover for protection and limit semi-permeable membrane outside of the cell, dividing the external media with the internal media of the cell, is the plasma membrane. Then we have here the core, the central, the control center is going to be the nucleus and the nucleolus here. And the matrix, the fluid matrix, is going to be this one, is the cytoplasm. It's containing fluid, which is called cytosol, and some organelles. We can see here a mitochondria, you can see here some vesicles, you can see here endoplasmic reticulum, we can see here Golgi apparatus. Some of these are organelles, solid parts of the cytoplasm. You can check here every single structure that we have in our cells, and composite cells or somatic cells actually, and you can check here the different kind of organelles that we have in our cells and their functions. As I mentioned, the plasma membrane is going to be separating the inside of the cell from the outside of the cell. And it's made mainly from, it's made of phospholipid bilayer. It's lipids. If you remember the classification for lipids, it's going to be triglycerides are the most frequent ones. And then you have phospholipids, and then you have steroids. And this phospholipid bilayer has two poles. Each phospholipid has a head and has a tail. And if you remember the composition, the heads are hydrophilic, they are water lovers, and the tails are hydrophobic or water feeding. Then inside of that bilayer, they have integral proteins attached, and they can work as a receptor, channels, or carriers. And we have glycoproteins and glycolipids. Then we have their phospholipids, we have their proteins, we have cholesterol as well, and we have embedded some carbohydrates, marking the cells as belonging to a particular individual. This is the graphical representation for the membrane, and you can see here this is the inside of the cell, this is the outside of the cell, and this is the membrane separating both of them. It's made mainly of phospholipids, as I told you, and this is the heads, and these are hydrophilic, and the inner part is made of tails. It's hydrophobic. And you have some proteins embedded in here, and you have as well some other molecules such as proteins and carbohydrates and cholesterol molecules embedded in the membrane. Again, this is a graphical representation for the plasma membrane. Other part of the cell is going to be the nucleus. And the nucleus stores the genetic information. It contains chromatin, which is condensing into chromosomes later, prior to the cell division. Then genes composed of DNA make up the chromosomes, and they code for the production of proteins in the cytoplasm. And it has the nucleolus, which is the dark staining bodies inside of the nucleus, and they're containing RNA and proteins, because it's the site for ribosome production. Here the graphical representation for the nucleus, and you can see here the nuclear envelope. Outside it has pores, and you can see here the DNA organized as chromatin. It's swimming freely inside of the nucleus, and you have here the central structure called the nucleolus, and you can see here the ribosomes inside and the proteins, because remember, this is going to be the site for ribosome production in the cell. Now we can talk about the organelles in the cytoplasm, and one of them is going to be the ribosomes. Ribosomes are small organelles where protein synthesis is happening. This is the main function of the ribosomes. Then they are freely in the cytoplasm, or they could be attached as well in the endoplasmic reticulum. That is called endoplasmic reticulum, the rough one, the rough rough endoplasmic reticulum. And the function again is going to be the protein synthesis. Then we have another type of organelles. Among them, the endoplasmic reticulum, it could be smooth or rough, the Golgi apparatus, lysosomes, and vesicles, and all of them together are forming the endomembrane system. Then one of them is going to be the endoplasmic reticulum. It's continuous with the outer membrane of the nuclear envelope. It is a system with channels and sacules. Again, it's two kinds. One of them is going to be the rough ER. It's called rough because it has attached ribosomes. And the synthesis of proteins is going to be the function of it. The other one is smooth. Then it's the smooth ER or endoplasmic reticulum, and it has no attached ribosomes, and the function is going to be the synthesis of lipids, and it has a function of detoxification as well. Then, as you can see here, in this graphical representation, we have the continuation of the nuclear envelope. It's going to be the rough ER, and it's rough because it has ribosomes attached, and again it's the site for protein synthesis. And you have another one, kind of endoplasmic reticulum with no ribosomes attached. It's called smooth ER, and it's going to be the site for lipid production, phospholipid production, and it's going to be the site for detoxification of the cell. Another member of the endomembrane system is going to be the Golgi apparatus, and it's again more membranes, more sacules. It's part of the endomembrane system, and it's containing enzymes that modify the proteins and lipids. Actually, it's going to be the place to storage some of the proteins produced in the cell. And another function is going to be the production of lysosomes. By the way, these ones are the trash can, the garbage disposal of the cell. In this graphical representation, you can check here in the center in green the Golgi apparatus, and it's going to be related with the production of vesicles in order to storage the proteins produced by other organelles. And it's going to be the site for lysosome production as well. Again, it's going to be the garbage disposal, one of the garbage disposal of the cells. And one of these garbage disposals of the cell, again, is going to be the lysosomes. They are containing hydrolytic digestive enzymes there, inside of them, and they digest or destroy some debris, important debris or worn out organelles inside of the cell. And other organelles working as a garbage disposal is going to be the peroxisomes, but they have inside of them some peroxidases, are enzymes to digest specific substances such as drugs, alcohol, and other toxins. And we have some amount of them, some increased amount of them, for example, in the liver and kidneys, because they are filter and detoxification organs in our bodies. And another kind of organelles are vacuoles. They are able to isolate substances captured inside of the cell. Now we're going to talk a little bit about the mitochondria. Maybe it's the only organelle you remember from the primary school. It's going to be the powerhouse of the cell. It's an inner membrane folds system in order to produce energy starting from glucose and oxygen, and it's going to be the site for cellular respiration. It's the site for energy production. And here we have a graphical representation of the mitochondria. Again, it's going to be the site for cellular respiration. It's going to be the site for ATP production, starting with glucose and oxygen. We have some other macromolecules maintaining the shape and the structure of the cell. It's going to be microtubules, intermediate filaments, and active filaments. They're going to be, again, part of the cytoskeleton in order to maintain the shape and the structure of the cell. Then we have a couple of organelles called centrioles, and you're going to remember them because they're going to be close to the nucleus, and they're going to be really important, especially during the cellular division, because they're going to be related with the spindle fibers production. And they are forming the basal body as the anchor point for cilium and flagella. And these two organelles are for movement of the cell. Yes, these two are the only organelles in charge of the movement of the cell. Cilia are heart-like projections, and they are especially in the respiratory system, and they are moving always dust particles and mucus, and that's the reason smokers are able to be coughing, because we're paralyzing the cilia because of the smoking habit. Another organelle for movement is going to be the flagellum, which is a single whip-like extension for cell movement, and the only cell that we have in our body with a flagella is going to be the sperm. It's the only cell in a human body with a flagella. Here the graphical representation for these organelles. To the left side you have the cilia. It's going to be hair-like projections of the membrane moving the cell. I mentioned we have those in the respiratory system moving dust and mucus. On your right you have the sperms, and the long organelle in order to move that cell is going to be the flagella. In this slide, you have an important resource. It's going to be the table with the structure in human cells, the organelles of the parts and organelles of the cell. And you can have here the names and the functions. It's going to be really important specifically for your evaluation. Pay attention and check this table 3.1 because it's going to be very useful for your evaluation. Going to the next learning outcome, we need to talk about the movements through the membrane. You remember the plasma membrane is selectively permeable, and it's going to be permeable for some substances going into the cell and some substances going out of the cell. Then we're going to talk a little bit about the transport through the membrane. Then regarding the transport, we have some processes requiring energy. These are active processes. And those which are not requiring energy are called passive processes. Then we have active transport requiring energy, and passive transport through the membrane doesn't require energy. The first of passive movement is going to be simple diffusion. And we're talking about the movement of solid particles from a place of higher concentration to a place of lower concentration. That is going to be following the concentration gradient. And again, it's a passive movement. It doesn't require energy. And we're going to see an example of that immediately. When we have a membrane, and we have a solution, again, it's going to be a solution, it's going to be a solvent, which is a liquid, and a solid, which is, in this case, a piece of crystal of dye, but it could be salt, it could be sugar, whatever you have solid, to be solved in water. Then there's going to be diffusion in order to distribute equally inside of the solution. That is called diffusion. By the way, the most frequent substances to diffuse in our body are gases, are oxygen, carbon dioxide, nitric oxide. All of these gases, they're going to be the main representation for simple diffusion. Now we have osmosis, another passive transport through the membrane. When we were talking about simple diffusion, we were talking about movement of solids or molecules. This time we're talking about the movement of water through the membrane. Then we have a membrane which is going to be permeable for water, but not for solids. And then this liquid, this fluid is going to be able to generate the osmotic pressure. It's going to be a force exerted on a selectively permeable membrane by the liquid. Then regarding the concentration of the solute in the solution, where we have our cells, we can have three kinds of solutions, and they could be isotonic, they could be hypotonic, and they could be hypertonic. The isotonic solutions, we have the same concentration of salts, specifically any solutes, but it could be salt. In the case of the blood, it's going to be 0.9% of sodium chloride, and we're going to have the same amount, the same concentration of solid outside and inside of the cell. When we have a hypo means lower concentration, hypotonic solution, it's going to be a lower concentration of solid outside of the cell. And eventually the water is going inside of the cell, it's moving inside of the cell, and eventually it's going to produce lysis of the cell. And the third solution is going to be the hypertonic. Hyper means higher, and we're going to have higher concentration outside of the cell, and eventually the water is going out of the cell, then it's going to be producing crenation of the cell. Then we're going to see an example in the next slide. Then you have here isotonic solutions, you have here hypotonic solutions, and you have here hypertonic solution. Then in the isotonic solution, we have the same amount of salts outside of the cell than inside of the cell. In the hypotonic solution, we have less salt in the out of the cell compartment than inside of the cell. In the hypertonic solution, we have more salt outside of the cell than inside of the cell. That's the reason we have no net movement in the isotonic solution. The water is not moving inside or outside of the cell, and we have regular shaped red blood cells. In the hypertonic solution, imagine the patient is dehydrated, then we have more salt outside and the water is moving out of the cell. Then eventually the cells should be crenated. And the reason we have in hypotonic solution, we have movement inside of the cell and eventually the cell is going to be lysed. Then we have another kind of passive movement requiring no energy, and it's called filtration, and it's going to be depending on the size of the molecule. It's not following a concentration gradient. It's not following the movement of water. This time it's following the size of the molecule. Then we're going to be moving liquid and solids from the high pressure to the low pressure and following again the size of the molecule. Another kind of passive transport is going to be the facilitated diffusion, and this one is facilitated by a protein carrier, but again it's not using energy at all. This one is using energy. It's transported by a carrier as well, but it's using energy. This one is going to be the active transport, and one example of it is going to be this potassium-sodium potassium pump, which is a very important mechanism that we have in order to get the energy and the restoration of the action potential. Then this sodium-potassium pump is going to be the best example of active transport. Again, active transport is using energy, and it's going to be a transport through the membrane using carriers. Here you have the graphical representation for active transport. It's going to be for the sodium-potassium pump, and it's going to be moving three atoms of sodium outside of the cell and getting two atoms of potassium inside of the cell in order to recover the negative potential outside of the membrane. And we're going to be talking about that during the chapter for cardiac cell, and we're going to be talking about the action potential in nerve cells later. Another kind of movement using energy, active transport, is going to be endocytosis and exocytosis. Endo means inside of the cell, bringing into the cell, and exocytosis is taking out of the cell. It's out of the cell. Then in endocytosis we have two versions, two flavors. One is going to be when the cell is engulfing something solid. It's ingesting something. Think about the cell is eating. It is called phagocytosis. Instead of that, when the cell is taking in something liquid, a droplet of liquid, it's going to be pinocytosis. And then imagine the cell is drinking something. On the other hand, when we have exocytosis, we're taking something out of the cell, and it's going to be vesicle fusioning with the plasma membrane, and it's going to be secreting something outside of the cell. This is table number 3.2. Please be aware, these are the mechanisms of transport through the membrane. We have passive methods using no energy, and we have active methods using energy. Be aware of the requirements and the direction of each of them, because this piece is going to be really, really important for the evaluation. Be aware of that, guys. The third important learning outcome in the cell level of life is going to be the cell cycle. And the cell cycle is the lifespan of the cell. It's consisting in a series of stages. It's controlled by the internal and external signals, such as the growth factors. Then we're going to be talking about the cell cycle. The cell cycle, including the division of the cell, it's going to be a series of stages that take place between the regular time and the cell division. And another important concept here is going to be the cell death schedule. Expected cell death is going to be called apoptosis, and it's going to be occurring at a restriction checkpoint if the cell did not complete the mitosis, and the mitosis is going to be abnormal. Then this one is an expected process to control the cell division, and eventually we're going to be dividing our cells, but some specific specialized cells no longer go through the cell cycle. For example, muscle cells and nerve cells, they have just a few divisions, and some of them, they have no more divisions. In a cell cycle, we can distinguish the regular time of functioning of any cell. Is called interphase. Actually, this one is about 90% of the time, and it's divided in 3 stages. Interphase is divided in G1, which is going to be the growing of the cell. S phase is going to be for growing and DNA replication, and G2 is going to be the growing the cell and preparation, especially with enzymes before the division. Then we have the nuclear division of the cell, which is called mitosis, and is divided in 4 phases: prophase, metaphase, anaphase, and telophase. And finally, we have the division of the cytoplasm, which is called cytokinesis. Cytokinesis is going to be the division of the cytoplasm. Then again, interphase is going to be the longer phase. It's going to be the most regular stage on the cell cycle. It's going to be about 90% of the time of the regular cell cycle, and it has 3 phases. G1 phase is going to be the one for growing. S phase is going to be for growing, and for DNA replication, then we have the chromatids divided into 2 sister chromatids. And finally, we have the G2 phase, which is going to be for growing and for creating the enzymes and final stages before the cellular division. Then during the interphase, we have 2 main events. They are the DNA replication and the protein synthesis. The DNA replication, again, is happening during the S phase, and it's resulting in 2 identical sister chromatids that form a chromosome. Then from chromatin, we're dividing our DNA, and eventually they're going to form a chromosome. The other important event during the interphase is going to be the protein synthesis. Then the protein synthesis, it's developed because of 2 processes. One is going to be happening inside of the nucleus, which is called transcription. Then we're using RNA in order to copy the genetic code from the DNA. And the other process is going to be happening in the cytoplasm. That is involving other kind of RNA. It's involving the messenger RNA, the ribosomal RNA, and the transferred RNA. And this one is going to be the moment when the RNA is giving the information to the ribosome, and in order to concatenate the several amino acids in a polypeptide, and it's going to be the origin for the protein. Then our genetic code usually is going in form of triplet code. Actually, it's called codon, and it's going to be a group of 3 bases in DNA and RNA, and they are transporting a particular specific amino acid. Then 3 of these bases are coding a specific amino acid. Here you have the graphical representation for the protein synthesis. As I told you, the first process is called transcription, and it's happening inside of the nucleus. We're replicating our DNA. We're duplicating our DNA using mRNA chain in order to get the information. And then after that, we're moving that RNA messenger outside of the nucleus. Is happening inside of the cytoplasm, and we're getting that RNA chain into the ribosome, and the ribosome is going to be using the ribosomal RNA in order to concatenate these codons but instead of having this information, it's going to be brought by transfer RNA, which is bringing the amino acid inside of the ribosome. Then eventually this is going to be the whole process. Transcription is happening inside of the nucleus. Translation is happening outside of the nucleus. Transcription is going to be the duplication of the information using messenger RNA. And outside translation is going to be the concatenation of several amino acids in order to get finally a polypeptide in a form of a protein. You have here 4 important elements of the DNA replication. Then you have the RNA polymerase, which is an enzyme in charge of forming the RNA molecule. And then you have the 3 kinds of RNA that we have. The first one is going to be the messenger RNA. It's going to be for coding the proteins, getting the code from the DNA inside of the nucleus. And then we have moving this mRNA to the cytoplasm. And inside of the cytoplasm, we're going to have ribosomal RNA, which is making up the ribosomes. It's going to be building the proteins inside of the ribosomes. And finally, we have the transfer RNA, which is going to be moving the amino acids into the peptide chain, synthesizing the protein. 2 other important definitions in the protein synthesis are codon and anticodon. The codon is a sequence of 3 nucleotides in the copy of messenger RNA that codes for an amino acid in order to send the message to the tRNA in order to get that amino acid. And the tRNA is containing the anticodon, which is going to be a sequence of 3 nucleotides that binds of a codon with mRNA. Then these sequence of nucleotides are for the sequence of nucleotides in mRNA, and it's codifying the sequence of amino acids in the protein. In the cell cycle, the rest of the time after interphase, we're going to be dividing the cell, and it has 2 parts. The division of the nucleus is called mitosis, and the division of the cytoplasm is called cytokinesis. You have to be aware from a parental cell, from a mother cell, which is divided in mitosis, you're going to get 2 identical daughter cells at the end with the same amount of chromosomes. As you can notice, a somatic cell has 46 chromosomes, and each daughter cell is going to have 46 chromosomes. Then after interphase, the DNA has been replicated, and it's going to be 2 sister chromatids joining at a centromere. It's going to be a chromosome. For mitosis, we have 4 stages. The first one is going to be prophase, then we have metaphase, then we have anaphase, and finally we have telophase. The prophase is going to be, first of all, it's going to be destroying or eliminating this nuclear envelope, and from chromatin that we have, DNA circulating freely inside of the nucleus, we're going to transform and condense these chromatin in chromosomes. And eventually the centrioles, they're going to be addressing to the pole of the cell, and they're going to be producing some spindle fibers in order to attach the chromosomes. Then when you have already the chromosomes, they're going to be eventually aligned in the equator or the middle of the cell. This is the metaphase. And you can recognize that because every single chromosome is going to be aligned in the middle of the cell, attached to the centrioles. And later, they're going to be dividing both chromatids. It's going to be half of the chromosomes, and it's going to be pulling apart to the poles of each cell, and it's going to be the phase of anaphase. Then you have the chromatids pulling apart by the centrioles, and we're dividing. Eventually they're going to be the poles of each cell. And the telophase, where we are resynthesizing the nuclear envelope, and we're having here the simple chromatids, and eventually they're going to be becoming again in chromatin. And this cleavage is going to be the beginning of the division of the cytoplasm, which is the cytokinesis. Here you have the 4 phases of mitosis. Prophase, you're going to have the condensation of the chromatin in chromosomes. You're losing the nuclear membrane, and eventually they're going to be the centrioles emitting the spindle fibers attaching to the chromosomes. Then you have the second phase of the mitosis, going to be metaphase, when you have the chromosomes aligned in the equator, the middle of the midline of the cell. And they're attached, the chromatids are attached each other. Eventually they're going to be pulling apart by the pole of the cell with the centrioles attaching using the spindle fibers, and we are separating the chromatids, and it's going to be anaphase. And the 4th phase is going to be when we have the chromatids by themselves in both poles of the cells, and now we're reforming our nuclear membrane, and eventually the chromosomes, they're becoming again in chromatin. And you can see here the cytoplasm division, and it's going to be the fluorocleavage, and it's going to be the cytokinesis, which is going to be the cytoplasm division. Why is important this phase of mitosis, of the cellular division in somatic cells? Why? Because it's maintaining the chromosome number. You already know we have 23 pairs of chromosomes, and we have each cell is going to be genetically identical to the other cell. Remember, at the end of the mitosis from a parental cell, we have 2 identical daughter cells. And it's going to be important as well because we're using mitosis or cellular division for the growth and repair of multicellular organisms. And on the other side, we have the next procedure we're going to be studying later, maybe for the reproductive system. It's going to be meiosis, which is going to be the division of the cell, but this time for the sexual cells, not the somatic cells, but the sexual cells. That is the ova and the sperms in our body, the gametes, and they're going to be dividing, and at the end, instead of 2 daughter cells, they're going to be 4 daughter cells with the half of the number of chromosomes. Then eventually, from a somatic cells, from 46 chromosomes, we're going to get 4 haploid cells, only with 23 chromosomes, and they could be an ova or a sperm. They're going to be totally different each other. Not like mitosis, they are identical dotted cells. And this is going to be the end of this chapter, chapter number 3, the cellular level of organization, and that is going to be all the material. Please be aware, if you check these PowerPoint slides, be aware, go to your textbook again and check of these concepts because it's going to be very important for your evaluation. Thank you very much.